Hello and welcome to part 2 of the DNA Profiling Lab. My name is Jeffry Petracca and I'm an educator at the DNA Learning Center at Cold Spring Harbor Laboratory. Now, last time we took a look at forensic DNA profiling and we saw how repeated sequence at particular locations in a genome could be used to build a profile for an individual, a unique genetic profile for an individual. Now, in forensic science, that is really, really important, right? So if we were to identify some evidence at a crime scene, for example, and we wanted to associate it with a particular individual, we might want to construct a DNA profile in order to place that individual at that crime scene. Recall that last time, what we did was we extracted DNA in the hopes of amplifying a very particular location in our genome, specifically the D1S80 locus on chromosome 1 of the autosomal genome. And what we did was we carried out the part of the experiment where we essentially got our cheek cells, we concentrated those cells, and then we isolated and purified DNA from those cells. We then set up a PCR in order to amplify D1S80 specifically. And today, what we're going to do is part 2 of the experiment where we're going to try to visualize our DNA, visualize our D1S80 locus to get a better look at how many repeats that we have at this D1S80 locus. Now, as we'll see today, gel electrophoresis is not necessarily the most precise way to get an accurate number of repeats. Remember, each one of those VNTRs, those variable number of tandem repeats, consists of 16 nucleotides. And so that number is very important, as I said last time. You're going to want to remember that, more so for part 3 of our experiment, which we will take a look at next time when we're using our bioanalyzer DNA chip to get a more precise look at how many repeats that we have. But for now, we're basically going to be using a rough and ready technique known as gel electrophoresis in order to identify the different numbers of amplicons that we had in PCR. And remember that whether or not we had different numbers of repeats on one homolog of chromosome 1 or the other, our PCR will ensure that both of those homologs will be amplified. Those D1S80 locations will be amplified on both chromosomes, regardless of how big they are. That's a key point to understand. And so essentially, what we're going to do now is set up our gel electrophoresis. So let's take a look at what we have here in the lab. So I've gone ahead and already set up a gel for us. I basically took liquid hot agarose, which is basically a powder. I mixed it with a buffer, and then I warmed it up in the microwave in order to get it steaming and hot. It's very viscous. And what I did was I poured it into this. This is a casting tray. These blue things on the end here are meant to create a nice little mold for our gel to sit in. And this little white thing here is called a comb. This is meant to give us little depressions in the gel. Those are called wells. And the purpose of the wells is to separate our samples from one another so that we don't contaminate each sample, and so that we can get a better image of the amplicons that we see from our PCR reaction. Now this is done. This took 45 minutes or so, which is not long for PCR. And so we're ready to go. So hopefully whatever comes out of this machine here is just D1S80 amplicons. And we're going to tell exactly how many amplicons we have using gel electrophoresis. We may not be able to get a good idea of the size. We'll get an approximate idea, but not really a precise idea. And so we have to set up our gel chamber to prepare it for running our gel. So what I'm going to do is take off these little Jello molds here. I'm going to pull out the comb directly up just to keep it from damaging the gel. We don't want to break the wells, right? And so what we have here is a gel that has just a bunch of 8 little wells in it. Those are, again, separate depressions that we can use to separate our samples from one another. I'm going to take this and I'm going to place it in a gel electrophoresis chamber. Notice that we have black lines on these casting trays as well as in the gel box here. And so the black lines and the red lines correspond to anode and cathode respectively, negatively charged and positively charged and respectively. And so I'm going to lower my casting tray into the gel chamber, matching black to black and red to red. We want to make sure that the current is running in the proper direction. The whole idea of gel electrophoresis is that it will separate out DNA. And so basically, the longer the pieces of DNA, the harder time they're going to have of navigating through the gel itself as they get pulled through an electric field that gets set up in the box. And the shorter pieces will go a little bit further. So in order to conduct that electrical field through the box, we need to pour something in here that will conduct charge. And that's what I'm going to use this for. This is TBE buffer. And buffers are solutions of ions that will resist changes in pH. But for this purpose, we're simply pouring this buffer in here to act as a conductor. So I'm going to pour a little bit of buffer on one side of the gel here. The buffer will go over the top of the gel. Then I'll pour a little bit more on the other. And again, I'm not pouring so much buffer to bury the gel deep within the buffer. I want to pour just enough to cover the surface of the gel. Now, once this is done, let's get our samples out of our PCR machine here. And so as a reminder, I have a total of 3 samples here. I have one sample labeled J. This is the sample that I was extracted from my mouth last time using a saline mouthwash. I have another sample of mine, and then I have my a sample from a family member, my girlfriend actually, who which is meant to show you guys that you're going to see different results between both mine and my girlfriend's, whereas my sample should be exactly the same. That's the whole point of this, right? And so I'm going to load these 3 separate samples into my gel, but notice that these samples are clear. You can actually see right through the tube. The buffer also is clear. The gel is clear. So it's going to be pretty hard to see if I've accurately loaded my samples into the gel. So what I'm going to do is I'm going to combine my samples with just a little bit of loading dye. This is a blue sugar, essentially, that will help to weigh down our sample, and it will also help to give some color to our sample so that we can see what we're doing. Only thing is, we still have more stuff going on in the experiment, right? So part 3 of this experiment, we're going to take a look at a bioanalyzer chip. And we need some sample, some amplicon here to analyze. So what I'm going to do is, before I combine my loading dye here, I'm going to take a little bit of my amplicon, 5 microliters to be precise, and I'm going to set it aside so that we can work with that on part 3 of our experiment. So I'm going to start with my tube first. I'm going to basically just measure out 5 microliters of my sample, and I'm going to place it in another microfuge tube for later. I'm going to go right down to the bottom, and I'm going to just deposit that sample in the microfuge tube. Now, I'm going to make sure that I'm changing my tips every single time here, because I don't want to contaminate my samples by mixing them together. So here we go. I'm going to go ahead and take out another 5 microliters from my other sample. Place that in the appropriate tube. And my final sample, I'm going to get another 5 microliters. Okay. So now that that's done, what I'm going to do now is add 1 microliter of loading dye to each one of my samples. The remainder of my samples, that is. So there were 20 microliters in all in these little PCR tubes. That means there's 15 microliters left now. So I'm going to add 1 more microliter to each tube in order to, again, give my sample a little bit of color and weight so that we don't have as much trouble when we're trying to load into the gel here. Now, I'm going to do something called pipette to mix, which essentially means you use a pipette to do the mixing for you. And so I go to my first stop, and I basically take the solution up, back and forth, mixing it all together. And I have a nice blue solution in my little PCR tube here now. Okay, I'm going to change my tips again here because I don't want to mix my samples together. Incidentally, this loading dye is pretty nasty stuff. If it gets on you, it will stain your hands and your clothes. So if you ever happen to find yourself working with this, just make sure you are careful with it. You don't want to get it all over the place. It'll definitely ruin your clothes. Not going to hurt you or anything, but not something that you want to get all over yourself. So last but not least, we have our final sample. Okay, so now all of these tubes have about 16 microliters of liquid in them. So that's good. Now, in our gel, we also have another stain, actually. This is called gel red, and gel red is a DNA stain. So wherever it encounters DNA, it's going to essentially insert itself in between the base pairs, and it will fluoresce under a black light. That will allow us to actually visualize our DNA once we run our gel. So just keep in mind that that is still in there, and that's going to help us to actually see our gel. So let's go ahead and now load our sample. So I told you guys there's 16 microliters inside of these little tubes now. So what I'm going to do is I'm going to load 15 microliters into my gel here. And so when you're loading a gel, it's important that you are very careful and you maintain a sort of a stable hand. So I like to put my elbow on the table and I use my other hand to guide the tip of the pipette into the gel itself. So if I start with my first sample, the one that I extracted last time, it's labeled J, I'm going to extract, or take up rather, 15 microliters of my sample. I put my elbow on the table and then I use my other hand to guide the tip of the pipette. And I'm going to put this sample in the second well that is in our gel. And I'm just going right beneath the surface of the buffer, and then I'm going to press down to my first stop. I'm not going to go to my second stop because you have to understand that when you're loading a gel, if you put extra air into the well, it's going to blow the sample up and out into the buffer. It's going to waft away and you're not going to get any results. So be careful when you're doing this step. If you find yourself doing gel electrophoresis, just remember also to change your tips at this point. After all of this hard work, you do not want to mix these samples together. That would be bad. You will not be able to get a clean result, and it will kind of defeat the purpose of this experiment. So I'm loading my second sample once again, and I'm going to put this in the third well. And you might be asking yourself, why are you skipping the first well? Is there a reason for that? And yes, actually, what I want to do is I want to put something in this gel that will allow me to tell approximately how large each one of the amplicons is that we will see in here. Now, we'll take a look at what we expect to see momentarily, but understand that if you just run a sample in a gel, without any frame of reference, you're not going to be able to understand the size or interpret the size in any way. So what scientists often do, and what I'm about to do, is I'm going to load what's called a DNA ladder. And specifically, it's a 100 base pair ladder. So this little pink tube here has essentially a mixture of DNA of known sizes. And specifically, it will separate each one of the fragments in this tube here by 100 base pairs. So starting from 1,000, going all the way down to 100. So there's there are fragments in here that are 1,000 base pairs in length, 900, 800, 700, and so on. So I'm going to put 6 microliters of this 100 base pair ladder into the first well in our gel here. And this is going to, again, serve as our reference marker. Now, the reason that I'm putting less of it in here is just because you don't need a lot of it to achieve the desired effect. And depending on the concentration of our samples in our gel here, it's going to run at different lengths. So we want to make sure that we're not producing effects that we want to make sure that the concentrations are relatively the same across the board. And so now that my samples are loaded, I'm going to go ahead and plug in to this power supply over here. And I'm going to plug in black anode and red cathode to one port. And then I'm going to set the voltage. So the voltage is a measure of the electrical potential energy running through this box here. And I'm going to put it right up at around 140. And I'm going to run it for a time of, let's say, 30 minutes. Okay, and I'm going to Once that's all set, I'm going to press this little running man over here and the gel will start to run. And so now what's happening is the power supply is providing an electrical field to this box, and it is flowing through the box. There's wires on either end. And any charged particle like DNA, for example, is going to get caught up in that electric field and be dragged through the gel. And again, like I said, depending on how large the fragment is, the smaller the fragment, the further it will go through the gel, because it'll have a much easier time of navigating through the gel. So let's wait on this. And in the meantime, let's see what we expect to see for our results. While our gel electrophoresis is running, let's take a look at what we should expect to see for the outcome of our experiment here. So you'll notice that this looks very much like the gel that I showed you earlier. So you're looking directly down onto the gel from above. And over here, up on top here, this is where the black line was, or the anode was. So I placed the wells, remember, right at the anode, the negatively charged electrode, which should make sense to you because DNA is, of course, negatively charged. And we want to push the DNA through the gel so that it runs through the gel in this direction. And so larger pieces of DNA are going to get stuck closer to the wells, whereas smaller pieces are going to be able to wiggle through the agarose polysaccharide matrix in the gel a lot more easily. Now, for our experiment, we loaded the ladder into the first well here, and that's 100 base pair ladder, remember. So basically, our 100 base pair ladder is a reference marker that is a mixture of DNA fragments that will separate themselves out such that each fragment is 100 base pairs difference in size. And so we should have, therefore, 10 of these fragments. So we'll have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this one corresponding to the 1,000 base pair largest fragments, and this one corresponding to the 100 base pair smaller fragments. Now, the reason that the fragments, the banding pattern that we see here, looks like rectangles is because we loaded our sample in a rectangle already. So DNA of equal size is going to be migrating at equal rates. And therefore, if we loaded our sample into originally a heart-shaped well, it would migrate in a heart-shaped pattern. And so that's why we're seeing rectangles like that. But what do we expect to see for our samples? So let's take this from the reverse approach. Let's see that, let's assume that I know my genotype, and I don't actually, but let's assume that I know that I'm 1823. That's my genotype at D1S80. So if I'm 1823 and I loaded that sample, that amplicon from PCR right into that well there, what should we see for our results? So if my allele is 18 on one chromosome of one copy of chromosome 1. That means that each fragment is 16 base pairs in length. That means I'm going to have a total of 288. If my other allele is 23 base pairs in length, I'm going to do 23 times 16. And that means I will have a total of 368 base pairs. Now, what I didn't tell you before is that these amplicons actually have some flanking DNA that's not repeated sequence on either side of where the primer's bound. So upstream and downstream of where the primer's bound, there's some stuff that is not repeated regions. So we don't want to consider that in our calculation here. So it turns out that that's roughly amounts to about 149 base pairs of flanking material. So I'm going to add that to these sizes here. And again, I'm doing this in reverse kind of. I'm trying to show you guys that if you know the size going into it, you can predict where your fragments are going to wind up, or where your amplicons are going to wind up in your results. So with this information, the total size of the amplicon coming out of our PCR should be 437 and 517. So we should have two amplicons rather, one of 437 base pairs in length and one of 517. So therefore, on our gel, we should expect to see two bands. We should expect to see one between the 500 and 600 mark relative to the ladder. So we go 1,000, 900, 800, 700, 600, 500. So our 517 fragment is going to be like right over here. And our 437, by the same principle, is going to be down here somewhere. And so there you go. There you have it. We'll have two fragments, and they will roughly be in the locations that you see here. Now, unfortunately, I'm coming at this in a different approach, right? So if I tell you my genotype going into it, yeah, you could figure out what your amplicon size is going to be. But you guys are coming at it we're like coming at it from a different approach right now, right? We're coming at it from the approach of, I don't know what my amplicon size is. I don't know what the genotype is in the sample that I loaded into the well, right? So how do we figure this out? Well, if you notice, you really can't tell much from these fragments here. I mean, they're approximately where we would expect them to be for a fragment, or I should say a size of about 450 maybe, and 520 maybe, but you really can't tell precise numbers by looking at an agarose gel. So we need a more precise method, and that's where the DNA chip is going to come in. And this is actually one of the limitations of gel electrophoresis, or agarose gel electrophoresis. It's very cheap, it's quick, and it's a good way to check if your amplification was successful, but you're not going to be able to get precise estimates. So here's a sample of an actual gel from a student, like a class that came here actually in February of 2020. And we see the ladder right over here. That's looking pretty much like we expect. This student has two clear bands, right? So they definitely have two different size D1S80 regions. This student also has roughly two different size D1S80 regions. But if you notice, it's hard to tell, how many repeats difference is there between these two bands here? We don't really know, right? This student has one solid band. So does that person have the same number of repeats on each copy of D1S80? Or is it just a slightly numbered, like maybe one repeat difference? Well, it's kind of hard to tell. And that's my point here, is that it's very hard to resolve these precise differences. And when you're dealing with a forensic investigation, you really want to be precise. You can't leave it to chance. You have to be as precise as possible. And that's where that DNA chip will come in. And so we'll take a look at that in part 3. Okay, so let's take a look at our gel results. So in the time that we've been talking, our gel has gone ahead and run for about 30 minutes. And I'm going to go ahead and open up our electrophoresis chamber. I'm going to reach right into the buffer here, and I'm going to pull out our gel. Now, let's take a look at that. Now, what you should see here is that we basically just have a blue line and a purple line. So is that our DNA? No, right? Our DNA, we can't see our DNA. That's what that gel red is for. Remember, the gel red, like I mentioned earlier, it will bind to the DNA anywhere that it encounters it within the gel, and it will fluoresce under a black light. So we need a black light to illuminate this in order to actually see our results. So I happen to have one up here hooked up to the computer. So I'm going to go ahead and put it under that illuminator, that black light illuminator, and project it up on the board here so we can take a look. All right, so here we go. Let's take a look. So I'm going to go ahead and turn on the illuminator, and lo and behold, there you have it. So what we are seeing here, so here are my two samples. So these are the first two that I loaded. And so this sample and this sample are virtually identical. So you can basically see that there are two fragments. So I have two different size or two different number of repeats on each copy of my chromosome 1, but I don't know how many, especially because this ladder here is a little bit condensed. It's actually overexposed. I can't really see or discriminate among the different size fragments here, unfortunately. But I can tell you that I definitely have two different size fragments. My girlfriend, on the other hand, she almost exactly has the same genotype as I do. There's a little bit of difference in the bottom fragment here. It's not quite as low on my girlfriend's sample here, meaning that she may have a different number of repeats on at least one chromosome. So unfortunately, like I said, this is the limitation of gel electrophoresis. I can't really tell the difference between these amplicons. And but what I can tell you is that the D1S80 amplification was successful, and it looks great. So this is beautiful amplification. I have no doubt that we'll see some good results on our DNA chip. So there you have it. So that's the end of part 2 of our forensic DNA profiling lab. And we will take a look at a more accurate and precise methodology in the Agilent Bioanalyzer chip, or DNA chip for short, next time in part 3. And we'll be able to get a better idea of what these gel results mean. We'll actually put them side by side, and we'll get a better, have a better understanding of how big each one of these amplicons actually are. So thank you so much, and have a wonderful week. We will see you next time.